Hey there, thanks for joining us at the Rabbit Hole of Research, where you'll learn a few facts that you can impress your friends with at a party. Hey, welcome back to the Rabbit Hole of Research. Down here in the basement studio, you've got me, Joe.
You got Nick.
We've got Nick.
Georgia.
We've got Georgia. We're all crewed up. We have no, we're not all crewed up now.
We're missing Mary.
We're 3/4 crewed up now. This is old school here. Mary is not with us, but we do have a special guest. Would you like to introduce yourself?
I'm Erin C, and I'm a evolutionary ecologist, but now I am a middle school science teacher. I do a lot of science communication and interpretation for the general public.
Awesome, yeah.
Very cool.
We met at MarsCon in Virginia Beach, so we were on a panel. I think I moderated the panel we were on. I can't remember if that was the case.
Possible.
Yeah. Oh well, that
I
was
think we were on a couple panels together.
It feels forever ago, but always it's a fun time. So if you're in the Virginia Beach area, or if you're not, just take a trip over. It's usually pretty nice in January. So, but yeah, we're here. We're continuing our Spider-Man villain series, and
what's our episode today, Joe?
today we're going to be talking about, in particular, maybe a little bit of Scorpion and the other chimeras in the Spider-Man universe. So we've got an evolutionary biologist, and so yeah, we're kind of...
See, I figured this was going to be the Pokemon episode. No?
Yeah, that's... You're right. Pokemon also have a lot of chimeras or chimeras. I don't know, what's that, like a potato and patata?
Sure. I know the exact definition of the two of them.
Yeah, so I can, let me get into it. I have my little...
Oh, please.
Let's guide us in. So every cell in your body contains the same DNA. Your liver cells, eyes, brain, feet, skin, the same code, identical instructions. What makes them different is which instructions they're allowed to read. Cellular machinery silences some genes and amplify others, following their section of the blueprint. All the cells with their different blueprints come together and make us work. But what if your cells could ignore their blueprints and you could rewrite those designs? Sounds like science fiction or Handwavium, but it's actually not. Scientists have done it. They've deleted genes a pig needs to grow a pancreas, injected human stem cells into the gap, and coaxed a human organ to grow, using the pig's body as an incubator to create organs that could potentially be used for a transplant back into a human. And Spider-Man villains have been riffing on this technology unwittingly since 1963. A man fused with the scorpion traits specifically, and why? Because scorpions are natural predators of spiders, of course. A surgeon who lost his arm and decided lizard DNA was the answer to regenerate his lost limb. A Victorian geneticist who spent over a century building organisms assembled from the parts of multiple beings trying to guide evolution, and he named his creations chimeras. The Greek used the same word for creatures assembled from parts that weren't meant to go together, and they thought of them as monsters. But is that what they are? Or is that just what we call something before we understand it, before we learn to build it ourselves? And as Richard Feynman once wrote on his blackboard, what I cannot create, I do not understand.
Interesting. So if he couldn't make pasta, he'd be like, I don't understand this.
That's right, yes.
Well, he didn't say he had to make it well.
Right, yeah, he didn't say that at all.
This is witchcraft.
But he would probably be a villain in the Spider-Man universe.
I guess
that's
So Spider-Man is and I'm always afraid of the pronunciation. Can you say it again?
chimera
Chimera.
or chimera. I don't know. Aaron, do you have
I say chimera.
chimera? Yeah.
Chimera.
Yeah,
So Spider-Man is a chimera, right? Because he's part Spider-Man?
I
A
think
spider?
you're right.
look at
No.
No.
No, he is not
What?
a
Okay.
chimera.
that's right.
Oh.
Because it's not even possible, but his DNA was altered with venom. And it's not that he has another set of DNA
Okay.
in his body.
Even when
So
he became the man spider?
Yes. So it's technically like his DNA. It's not spider DNA.
Oh,
So
right. Yeah.
people think, or I was reading a little bit about it earlier today, and they think it's more like equivalent to a radiation mutation.
And that was one idea that it was radioactive. They say a radioactive spider bit them. So that would be
Radioactive
DNA
spider.
DNA kind of mutation
But the fact
versus.
that he's able to throw webs and
Well, that's
Well,
and
that-
his-
That's web shooters.
yeah.
oh, it's not. He isn't actually.
Well, it depends
Hey.
on the
I thought. I thought.
right.
Oh.
That one stems off a lot.
Yeah.
Okay,
So it's
we're
once
not
he
really
gets to
clear
man
on
spider,
that.
that's when he can do the webs from his body organically. Every other time he is creating that in a lab.
Okay.
So the chimera is an organism with cells from two or more genetically distinct organisms, not just DNA splice, but genuinely mixed cell type organisms. Is a rough definition. Is that fair to say, Erin? Or
Yep. I
Yeah.
was trying to come up with a good definition that would be understandable. So I wrote down an organism composed of cells from two or more distinct organisms.
Yep.
And that
So
could be
Jurassic
organisms of
Park?
the same species.
Would that be Jurassic Park then or no?
Yes, it would.
Yes.
Ah!
Yes, you would.
You
would.
got
Yeah.
me.
Yeah.
Yes.
Yes. Jurassic Park would, because we need to do a Jurassic Park episode because I still don't know why. And we're getting a little off track, so it's not Spider-Man related. But yeah, already. But I don't know why they use lizard or reptile DNA. Because even in the movie at the beginning, they go, oh, birds are the evolutionary cousins of dinosaurs. And then they go, oh, we couldn't have the whole sequence, so we used reptile DNA. So I think something nefarious was going on from the start. And because it really didn't make sense why they made this kind of weird jump. So it was like, so that's one of
seemed like
many problems.
They were pretty successful, though.
They
You know?
yes, a lot of Handwavium. Yes, they were. But yeah, that's all right.
Now,
That's
not
a little.
all of Spider-Man's villains fall in this category, right?
No, not all of them.
But, like, a
Scorpion.
good number of them?
Scorpion is one. The lizard probably is another.
I'm trying to
two.
remember what
Rhino.
the jackal was. What was his
I remember the
spliced
jackal.
with? Because I know he ends up having. He starts doing a lot of cloning. We're doing some pre talk about, and I can't remember. We'll have to go back to that one.
Yeah.
But
I know the other ones. Yeah. So Scorpion was, like I said, he was made. James.
Have another
I know.
glass.
It's a tongue twister there. I'm like, yes. He wanted to find a rival that matches Spider-Man. And so he was like, so a natural predator to spiders are scorpions. And he said, let's make this a scorpion hybrid, and tried to create that chimera so that you would have the scorpion with the tail. And
Yeah.
looks like you want to jump in there.
Yeah, I went on my own rabbit hole of research with that because I was like, okay, they said Jameson paid to
That's right.
have
Mm-hmm.
Scorpion participate in a human experimentation. And I come from an experimentation background. I know that Joe does as well. I don't know as much about you guys, but
No.
I was like, hold on. You don't pay to be a part of a human experiment. You get paid or you just volunteer. And I was like very excited. I was like, yeah, what I think is true. And I was like, how much did Jameson pay for this random man to join a human experimentation study? He paid $10,000.
Yeah.
And I was
Oh,
like, oh my God.
Yeah.
And that was, what was
60.
it, like 1960s?
65 or, I
Wow.
think 60. Yep.
What's the
Yeah.
inflation?
Well, you had, I mean,
really wanted to get the Menace Spider-Man.
they did move ahead though, right? Because at the Depression, it was a meal and a suit was the going rate to get experimented on in the reign of Superman. That was
It was
that was it.
hot meal
So,
and a
yeah.
suit.
that was Jonah Jameson.
Jonah Jameson.
Get it all straight there. Had that, and this was 65. That's before technology. So now one would probably lean on CRISPR because I think it was like gamma. It was like injecting DNA. So a lot of it was either we're just going to inject random DNA and then make something work, or we're going to use gamma radiation to alter your DNA and fuse. So it was,
I looked a little bit into that because I was like, okay, how did he even do this? And it was a mix of radiation and chemical therapy.
mhm.
And I was like, okay, does this even work? And so I started looking into it, and it really only works when you're starting to form
Mhm.
an embryo. And I was like, oh, so it wouldn't work anyway on a full-grown man
Right,
to
right,
do all
right.
that.
Yeah,
Wait,
I mean,
so I can't become the scorpion?
yeah.
What?
Well, the whole when you're in an adult,
You're in adult.
when you're an adult,
Oh,
when you're in your adulthood, or even in child, yeah, everything after embryo, your cells are reading their instructions and they're doing what they need to do. So to come in and to make modifications there becomes extremely difficult throughout the entire body.
because
So,
you would just fight things off as a foreign. Is that right? Or
well, not necessarily. How do you get all your cells now to adopt this new DNA and replace your original DNA functionally?
right?
Right. So how do you do that? So
when-
that's,
When you were talking about scientists that are doing things like this,
right.
you're talking about
Embryos generally, yep.
Yeah.
Yeah, embryos where you have fewer cells, you're earlier, so the cell paths have not been set. So a lot of the stuff with correcting genetic defects, a lot of that's been done in embryos, and so they go in and they use CRISPR. It's just the new technology
Can you
that can go in.
describe
And
what CRISPR is?
CRISPR, I guess, best is molecular scissors, so they can go in and they can clip out a region of DNA and then replace that with another piece of DNA that would encode a gene or a correction gene that would
That's
be there.
just
So
amazing. Would
yeah.
you say CRISPR's been around, what, 10 years or 5 years? Or
sheesh,
Do you
I
know?
don't- I think in the
They won a
mainstream.
Nobel
Yeah.
Oh, damn.
I just remember probably like,
I don't think so. It's like a lot of research and science. By the time it makes it to mainstream, there's been
right. I'm
many
curious.
years of, I want to say like the
and then,
90s.
and then
or,
also, when was it actually successfully used? You know what I mean? It was probably in
Development.
for quite some time.
Yeah.
So,
It won a Nobel Prize in chemistry in 2020.
okay.
Oh, wow.
I just remember, this is my background for CRISPR, listening to a Radio Lab episode.
I mean, that's a good
And it
place
was
to listen
really,
to.
it was really cool. It's like, whoa, but that was probably 6 years ago or something. I remember listening to that. So
Interesting. Yeah, I wouldn't have thought of when it came about. Because I know Joe has mentioned it before, but I just like thought it was a thing. It's always been a thing, right?
no, I knew it was pretty cutting edge.
Yes.
It is pretty amazing. I can't imagine how excited they would have been that worked.
So with CRISPR, is it? I don't know how to put it. So for CRISPR, do they do it for people and things that are like actively living, or only embryos?
Well, embryos are technically living.
Well, I mean, mature.
Can it be
Mature. Yeah, I mean,
can it
that's
be a later
so that's
development?
that's an interesting question.
They
No.
did it. They do it with bacteria, which are already capable of making their DNA as adults. So they attach to another bacteria and they exchange DNA, and then instead of reproducing with that exchange of DNA, they just change their own DNA, which is how they can adapt so quickly to antibiotics.
Right.
So and their DNA kind of forms a circle before they undergo their cell divisions. So it's a little easier.
And smaller genomes. But for a fully mature, developed organism, I think is the question. Yeah.
Yes.
Applying CRISPR, it's targeting to the place you want. And I don't remember the research, but was that the hearing defects? That was done in children, I believe.
Yeah,
but that was a very specific.
There was a really interesting case where someone wanted to reverse that and actually give a child hearing defect because they were in the deaf community and they wanted to have a child that they had in the womb. They're like, can I please have this child also be deaf? Because if they're not deaf, they wouldn't be part of my community, and it's a cultural thing. And that went up into the court systems. I don't know what the outcome of it was.
That is fascinating.
Wow.
Wow.
So
I
Maybe
was not
opposite.
expecting that.
Wow. Yeah.
I thought you misspoke, and I was going to be like, wait, you
Yeah,
want to
that is-
purposely make someone deaf? All right.
Yeah. Yeah. So you can start, kind of
who
these villains.
decides
Yeah.
what,
So you go
what
and,
is good and what is
Right.
bad at that point.
Right. But these are very targeted. You're not affecting whole organism. You just need to affect the cells that are involved in hearing.
Okay, so
you're-
that manitar, I know manitars are real, but I mean,
Sheesh, we went
would
from
that be
real world
Do
to
they share DNA, or do we ever never?
they're
or
all
we
one.
never.
It's one organism.
It's one
It's
organism.
one organism, right?
But it has
Right.
DNA of a horse and I don't know human.
And a human, I presume so. Yeah. Yes.
I thought it was just a specific
Yeah.
DNA that is horse human.
Horse human. Yeah, so the way it would work if I had the Manitar, wasn't
Sorry.
prepared for this.
Sorry.
But like any of these, I guess you would need. So what changes would physically need to happen? So that means that during development, there was some mix. You've included now a second or copies of the genome of a horse. So those genes are there to be turned on and off. And then once those genes are there to turn on and off, then you can have developmental programming go and turn those genes on and off. So for the rare horse butt, leg, hind legs, and midsection, those developmental genes would kick on, and then you would form that. And then in this soup of development, the human cells would then turn on and send their signals and, okay, all right, horse stop, cells stop growing, and let me develop now the human part of it. And so that's how you would think about it. But all of the DNA would be in each cell. So each cell would have horse and human DNA, and you're the way your cells are all Georgia cells and DNA, or Nick or Erin or Joe, they just turn on what they need to turn on developmentally. So as an embryo developing, the cells literally reach out into the space and start to lay the body plan. And as they lay down, they send signals to the other cells and say, okay, you guys, you're not doing this. You're not being feet. You now need to be the ankles. You need to be the legs,
Horse, and then
and you
could you
grow
have
out.
a manitar where it was a human, like a human legs?
Yes, isn't that Bo Jackman?
Oh,
No, isn't that what's his name? Bo something?
Will Arnett's character, BoJack Horseman.
BoJack Horseman, yeah.
Okay.
But yeah, that's how it worked. And it's interesting because let's go back to the real. I had mentioned in the open the example with the pig where they're trying to grow human organs in there. So what they do with that, they take CRISPR, they knock out the genes that would make, let's say, a pancreas or a heart, whatever organ you want to fully function, and you replace that with human genes so you can build in the pig, in the embryo. So you functionally remove something. So don't make a pig heart, and you replace that with a human DNA. You said make a human heart, and then you raise the pig up, and it starts making a human heart. So in human research like that, they usually have to stop at day 28 or something early. You're not allowed to do genetic work or chimeric work on embryos, and so they stop it. But they can see the new organ starting to form. But they do this in mouse and rat, so two different species. And you go, okay, so knock out the mouse, let's add rat, and then grow a pancreas. One of the interesting things is that because you just told the cells the mouse just to grow the pancreas, the rat pancreas, you got to remember organs are very complex. And so where do you get all the other genes and structures from? The arteries, veins that deliver blood and take away the used deoxygenated blood. They find that those are actually still mouse. So the host organisms cells. So when you have it,
but they're doing stuff they wouldn't normally do.
Well, they normally would make a pancreas. So it would be a mouse, 100% mouse pancreas. So that's because you tell it to do the rat pancreas. But it would have pig
might need different
veins in it
structures
and
things.
Right.
So that was one problem I was finding with implanting human tumors in mice, was that eventually the stroma, which is that membrane that attaches the organs to the rest of the body and the blood vessels that attach to it, those are still mice stroma,
right.
And they will eventually change the behavior and the drug response of the tumor.
Yeah.
So there are some effects.
So there's one other layer I should mention maybe as we get into this. Okay, go for it.
So if you're growing like a human heart in a pig,
yes,
wouldn't that have, I don't know, side effects for said pig?
Well,
like I'm assuming.
Well, I don't think
I mean, that's a different,
I don't think the pig's going to come out of this alive.
The heart would still function normally, as normal can be for a pig with a human heart.
Up until the point
That's when they need to take the heart out and put it into a person, right?
Yeah,
that's right. Yeah,
I was just saying, having that in a pig, like pumping the blood,
right,
is a different muscle than what it would be used.
Pigs and humans have very similar organs.
Physiology. Yeah.
That's why we
Oh.
use.
Okay. I
And
was
you're
like,
using the
And
pig.
size-wise, are
Right.
you thinking
Yeah.
more like size-wise?
Yeah.
Yeah.
I think if the heart,
size and structure.
yeah, and the heart's a muscle, and this is where I was going to say the second layer, because what the other thing that happens is something called epigenetics. And I know I mentioned this on other episodes. Epigenetics is a feature.
Cruel mistress.
That your genetics is a cruel mistress, right? And so we have the instructions of the cell. I was just saying that, what gets turned on and turned off. So you have those instructions, but you also have then epigenetic kind of overlaying that. And so that also then can add an extra layer of information to tell certain genes to turn on more, to turn on less, and you can inherit these kind of factors. And so you go, and so any your environment can cause certain genes to be turned off or turned on. So when you're growing these chimeras and you're trying to do that, not only do you have the situation where you have the cell types, because you didn't replace all the genes, so you also have the hosts actually imparting its instructions on that new DNA. So we were saying about size, you could have a case where in the pig, there is instructions already, epigenetic instructions, that even though you have replaced this, making a human heart, it would probably form it like it would in a pig. And to Erin's point, we use pig because that heart and size is probably close to, you would grow the pig to a certain size. If you need a heart for a child, you would have a little pig. If you want an adult, you would grow it bigger. But the pig probably would override and make the organ into what it needs. So if you grew it to maturity, the heart probably would function like a normal pig. It probably wouldn't know the difference, because a heart cell in a pig, a human, a dog, they're identical structure and function.
Yeah. Like
So-
the amount of beating, like the time.
Right. That would be, I think the Epics overlay would start to control that.
Yeah, they did a lot. This is old research now, but with Drosophila, the fruit fly, they moved different genes around and they put genes from other animals into the fruit fly to see what would happen. So they put a cockroach leg gene on the fruit fly's back and replaced it with a wing.
Yeah. Mm-hmm.
But what grew was not a cockroach leg. What grew was a fruit fly leg.
Yeah.
In that spot where they said, hey, make a leg.
Oh, wow.
Yeah.
So even though it's from a different animal, those instructions, it's going to be like, well, I am a fly. So you gave me a leg gene. I am making a leg.
interesting.
Yeah. But you do have, there's the pictures where they can grow human ears on the backs
Okay,
of
so
mice.
So the
so that's
example
really cool.
of the fly.
Yep.
Jeff Goldblum.
Yeah, that's a chimera.
That is a.
Yes,
even-
it got
even-
mixed. Yep, the
DNA
DNA, human
got
DNA,
mixed.
and fly DNA got mixed
It was
together.
only the head that was.
That's
Jeff
the original. In the Cronenberg '86 version, Jeff Goldblum,
Oh,
he
it
turned
does?
into a complete fly. Remember?
Mhm.
Yeah.
Oh, it's just the Vincent Price.
The Vincent Price
that's
one was
the
a hit. Like,
head.
help me!
That's actually hilarious because I was talking to seventh graders about fly today as we talked about
Awesome.
meiosis. And they were like, oh, could this happen? And I was like, one, I'm so proud of you for knowing what the fly is. But two, you can't
Yeah.
breed them together because the chromosome number is different. So
Mhm,
I don't
mhm.
know how
Mhm.
that would work for adding in fly genes where you have the head is a fly. If your
Right.
chromosome number is different, like how are you splicing those genes in
Yeah.
and what's being turned on and what's being expressed? And I don't know. That
Yeah.
would be more into your ball court, Joe.
Yeah, that's make it work. Yeah, you're right. We didn't mention with the Minotaur. That was something also. Where do these new horse genes go? And you could make it where you add chromosomes in. So you wholesale in chromosomes. So you could have multiple sets of, and this happens with fraternal twins, I believe, that during development, they just take in. So they have two sets of genomic DNA, and then they're essentially a chimera of their twin. And so they actually have both copies of DNA in there. So you can have multiple copies, and there's organisms like Lily, I think they can have many copies of their genome. So it's not metabolically taxing to have more DNA. The problem is when you get to cell division and things like that. These are finely calibrated systems over millennia of evolution. And so when you're trying to divide the cell, your machinery is expecting a certain number of chromosomes. And now if you got more, then you will start to get mismatches where some chromosomes are left behind or move to different places. Then you would develop mutations and probably death. And so probably a lot of these early embryo, you try to do it in embryo, you lose a lot of them because of these issues that they didn't really, this didn't work
they're
out so
just
well.
like, this is
Right.
not the way it's supposed
Exactly,
to be.
yeah.
So what about with the horse and donkey, the Ginny? Are they
Yeah.
just so similar that having them,
Reproduce.
yes,
Yeah,
without using any
Horse and donkey makes a mule. Yes.
mule? Yeah.
Yeah.
A
You say
Jenny?
A
I
Jenny.
thought it was a Ginny.
I've never
No, I
heard
never-
that.
I was like, do you it's like a pet or something?
It might have been someone's pet. I don't know. Pretty sure it's
I was like, I'm like, what are you talking? A Jenny? What is that? It might be some term for a female donkey. I don't know. Okay. Yeah, they're just really close in two close species, but they are sterile. You know,
So then
they
the
can't
mule
reproduce.
will
Yep,
not
the mule cannot
reproduce.
reproduce. No. So you go, and I think it's a mismatch. I think the chromosome number is close enough that you can,
Oh, interesting.
and they all can line up properly that you will get appropriate crossing. But yeah, the same thing in some flower species, plant species. If they're close relatives, they can do it. Their chromosome number, like Erin pointed out, that's close enough that they can actually, everything can line up, and then you can get division, and so you can actually make the appropriate cell division and grow into a plant. But usually they're sterile or other issues crop up. They don't make it to adulthood or certain things like that. But yeah,
so doing it in CRISPR and stuff. Would
Yeah.
something be able
Yeah.
to reproduce or no?
that's a good question. No, you probably could. If you really wanted it, could you try to add back in the
The
genes that were
reproductive.
taken out, the reproductive genes? You might be able to. Yeah. Right. I don't know if you should, but
Well, I don't think
know.
you should.
They said that
We're
the
talking-
dinosaurs wouldn't reproduce.
That's because they use reptile DNA.
I teach a class on the evolution of dinosaurs in the summer, and when we get to my genetics unit, we talk about Jurassic Park, how that can't happen and why it can't happen, and we look at then a Paleocene park or Pleistocene park. Sorry, so looking at mammoths and things, but the dinosaurs, we don't know exactly how they reproduced. We would have to use their closest living relatives, birds, to figure it out, or just make assumptions, inferences.
What were your assumptions?
The assumptions are that they court like birds do with
Mhm.
displays to determine physical fitness. That might be like, you got to dance for a long time. If you dance for a long time, you dance for the longest. You obviously are physically fit more so than the person who had to tap out in 5 minutes.
I would love to see a T-Rex dance like that.
T-Rex dance.
And then the feathers or the skin color, the more
Mhm,
vibrant
right,
they
that's
are,
right.
the better they are at avoiding predators. Or that is even an indicator of how well fed they are, if they can get certain nutrients that would create those bright colors. So those are what birds do today.
But I was going to say to Nick, your point about trying to modify and use CRISPR this way, I think someone who was ahead of the game is Mr. Sinister, because he had the Chimera program where he was trying to create these he was trying to force human evolution. And so he created and used his wealth from Victorian era into modern times and tried to take superpowers from other mutants and then recombine them
So
into
kind of like a Frankenstein.
exactly like like a Frankenstein,
A
except
genetic
a
Frankenstein.
genetic Frankenstein, right? That would do that, yeah. So because Frankenstein was a chimera at some level also because I think they used animal parts in there. I think it was like not. I don't think it was 100% human. I think they had I think Dr. Frankenstein had to get some bits, so it'd be Frankenstein's monster. But yeah, Dr. Frankenstein. But yeah,
Frankenstein.
Frankenstein.
Maybe they were friends because that sounds like about the same time period. Do you think
You're
Mr.
right. Yeah, they could. yes.
Maybe they
You're
were
in there.
like buds.
Yeah.
What
But
was it?
your
about
body does reject things even from your same species
Mm.
if you don't have the same blood types.
Yep. That's right.
So
That's right.
Frankenstein's monster.
Yeah.
Like,
Yeah.
he would have had to figure out, okay, what blood type did this body have? What blood type did this body have? I don't
yes.
think they had
would
discovered
have
at that
been-
point yet.
On that, like if something If something is completely drained of blood, does it have a blood type still, or can you just start pumping whatever in there?
Well, your T cells, B cells, I don't know if they'd already begun then too, but you still have an immune response.
Yeah.
And what you're really looking at is the antigens on the outside of your cell. They're like keyholes on a door. And you have your own antigens and they recognize each other. And then if your antigens don't unlock the door, then you're not allowed in. You're not allowed in this body. They need to kick you out and destroy you. So your whole immune system kicks in. And that's when you get limb rejection or tissue rejection if you've had a skin graft. You can even have rejection of your own skin. That's the whole thing.
Wait, what? What was that?
Yeah. If you have something happens to your face, say a monkey bit you or you have missing face, you might have a part of your eye taken and then regrafted onto your face so that you have that skin there. What can happen is your body either rejects it or that's not supposed to be there. I'm not taking that. Or it can absorb it completely where it's just all that. I'll just take that back into the body. And then suddenly you don't have it there anymore either. That you just wasted part of your leg up on your face. But it's a gamble. You can do certain things to try and help your body accept it. But ultimately it's up to your body.
Yeah.
It's going on.
Weird.
So as you develop, early in development, you develop self. And so your cells know self. And so they won't respond to you.
For this,
That's why, like with cancer, it's so hard to treat because your body sees that as self, even though it's now cell division now is it's just there's a genetic mutation generally, and that's causing kind of a runaway cell proliferation. And so you have this kind of issue, but your body's like, this is us. We got to live with it. And that's why it becomes a lot of the work is to actually use it against itself and say, well, if it's growing so fast, can you put signals out that will tell your body, I think this is wrong. And so go start to attack it and clean it up or to get rid of it. But yeah, that's why cancer is such a hard disease.
So when you grow the human heart in the pig, what are the chances of that to be successfully transplanted? You know what I mean? Have they had really good success rate of that? Or
One guy they did. Yeah. I don't know.
But I think that was
how the follow through has been going though.
Was that a pig heart though?
I think it was in the last few years.
They haven't. I don't know if they've grown a fully mature organ yet. Human
so this
organ
is something more that they're just experimenting with, and it isn't really a practice.
Because in just rules, ethical considerations, it's embryo research especially in America is pretty limited. So you can't do a lot of embryo research, especially with human cells and human kind of work past a certain day in development. And so it's like 28, 30, I forget, it's really early on in development. But you can see a lot. You can say, okay, you are starting, the cells are differentiating and are forming.
Are they doing that?
I don't know. Sure, maybe, hopefully not.
I did a quick Google search. There have been 2 people who have gotten a pig heart transplant, but it is the actual pig heart.
Right.
Because they were not eligible for a human heart transplant. Don't know why. There was one in 2022 and then one in 2023. Yep. The person in 2022 only lived for 2 months after this transplant, and it was linked to possibly a virus that was from the pig. And then the other person survived for 6 weeks after the transplant.
Oh.
Yeah. Even though
these are not high.
Died, these not great. Definitely better to have a human heart transplant. However, I guess they were working with what they could. And it's still good for science to be cool. Well, now we know what can go wrong and what can go right, but not great for.
Right.
But then I think
just
there was
fact that it worked for 6 weeks.
But you had, I think it was in China recently. They had they used a pig liver temporarily. So this kind of xenotransplant. So they used the liver temporarily instead of using external equipment. They connected a pig liver up until a human liver became available, I think, and then they transplanted.
And how did that
I think that
out?
went well. Yeah, I think that person was living and.
How long did it last?
What's that?
Or you mean how long was the temporary?
Yeah.
Yeah, I think it was. Yeah, I don't know. I can't remember. Weeks or something like that. Yeah, it was in lieu of external equipment, kind of filtering blood, right? Because your liver is filtering your blood out to pass to your body. So this is instead of having there's external equipment, and that's been tested,
And is
but
that
this is the
like
first.
a liver would be easier to do that than a heart?
I don't know if it's easier, but yeah.
Like maybe the function of a liver wouldn't be as...
Yeah, I know. I think
I
it's
don't
still...
know.
I think there's still this kind of issues with that because you're putting the pig heart in. The idea of trying to do it with human, and particularly the person who needs to transplant to use their cells, is that, as Erin was explaining, all of these kind of cell factors and external kind of lock and key, search and destroy things would be removed because now this is you, essentially. But that gets into this other part I was saying, that when you do it that way, you still have all pig. I think the one study I saw, it's one human cell per 100,000 pig cells. So it's really like a low, is it really human? Or is it mostly, it's not even mostly human, it's mostly pig. Because a lot of the other superstructure that makes the organ, the veins, the arteries, they are all from pig. And so the epigenetics is all pig. So you have this kind of, yes, it's human. It was seeded with human cells. You're trying to grow a human-like heart, but it's not. So yeah, I think there's a long way to go. It's not like this is where the field is headed, especially when you talk about chimeras. That's
all right. Hypothetical. All right. So say that I want to have a liver just a liver ready for me.
Right.
Would I be able to go ahead and be like, hey, here are my cells. Can you put this in a pig and grow this for me? Would it?
You look that up in the phone book?
Who do I call? To the island of Dr. Moreau? What are we doing?
Somebody played around with this. It's a fictional concept, a children's book called Scorpion House.
Oh.
And I read it when I was in high school. But the concept is a young boy who had a lot of siblings. They guessed they were adopted. They lived in this old man's house. Suddenly, every so often, his siblings disappear. And then he figured out, oh, we're all clones.
Mm-hmm.
And we're just, as this old man needs organs, he's taking them from us. It was a really interesting concept. And like, hypothetically, probably, because it's genetically identical to the old man, his organs wouldn't be rejected by that body. But of course, there's always a chance that it would be a very low likelihood of that happening. I know that they are trying to grow organs in labs. I don't think that is financially sustainable because you have to grow like a specific person's organ if they wanted to like have an organ bank for just themselves to make sure they could live longer, because that also doesn't guarantee a longer life anyway. That's just, oh, you have one specific organ that fails. Luckily, you have 6 of these cloned and growing in this lab. And that like you have other issues with growing old, like your telomeres an issue.
Yeah, and then growing organs in a lab is also, once again, they're complex 3-dimensional structures. And so how do you get that to grow without all the other information that's feeding in? So as your body's developing, like I said, there's a bunch of other cells coming together, giving a bunch of kind of
and it
directional
all has
cues.
To work right and work together.
It has to work right. But if you're in a cell, you just got a pile of cells. Those are, you go, I want a heart. So if you just take heart cells and you try to grow a heart, you just get a mass of
I
heart
have a hard
cells.
time growing a turnip. Come on.
But you need all the other cells. Like I keep mentioning veins and arteries. Like there's a bunch of other connective tissue in there. There's a bunch of other cells that form the structure as we know it's a heart. So I think that's also, how do you make the scaffolding? So they're trying to make 3D print it with biomaterials that you can seed with different cell types that then they may talk to each other and send the right signal and form the organ. So forming a whole organ is difficult. So really it's probably easier to clone the person.
Hypothetically, no.
Clone a person. So you would clone, it would be little mini nicks running around, and then you would just off them when you need them.
Can I just keep them in like test tubes or something?
There's also a book, Never Let Me Go, by Kazuo Ishiguro. And so the premise was similar, where the clones, they don't realize that they're clones of other people, but they actually have jobs and they go out, they do things, and then they're, they kind of. Is
that what doppelgangers are? Am I a clone?
You're really a clone. That's right.
I knew it.
But remember that children's book about the
old McDonald's?
Yeah, he made like a dog boy.
Yes,
and
those are all chimeras. Yeah, yes. Yeah, was it Old McDonald's Farm? No, it's Genetic
Farm. We probably have it over
I was
there.
actually looking. I was seeing if I could lay my eyes on it really fast. But yeah, No, that was fun.
It kind of creepy.
Yeah.
But...
Yeah.
So I do think that going back to Spider-Man stuff, there are different iterations where more of the villains are chimeras. Like,
we have a tarantula, rhino, scorpion.
That's just the thing.
And
some of them, they are more mechanical, where others,
like Doc Ock.
Doc Ock is mechanical.
Doc Ock's mechanical.
Yeah, he's not.
Yeah.
He's not octopus.
Yeah.
Not at all. No.
No octopus.
Breaking here. Doc Ock is not an octopus.
But that's like the scorpion, though, too, because in some iterations, he is a chimera. It was a genetic kind of DNA, radiation, chemical mutagenesis, DNA. But then in another iteration, I think like in the movies, he's like mechanical.
Yep.
Like he has a mechanical suit. It's more technology based. They wanted to get away from the Handwavium of,
maybe they just wanted to, they thought this is fun to draw.
Yeah, I guess.
I
Or
mean, yeah.
you want to explain it to science. You're like, let's get to science right.
I think maybe they wanted to do the drawing first and then, okay, can
mechanical
we explain
suit.
this?
Well, maybe that's, well, you also had a lot of these comics, they were done Spider-Man in the 60s. And so you had fear of other communism,
Fear
immigrants.
Of science, maybe?
You had at that time, you're right, the atomic age was in full swing. Mechanical DNA was just starting to be understood as this kind of hereditary kind of thing, manipulation of that. So you really did have this, and this was being shown up in comics as you have across, we're picking on Spider-Man a little bit because the Spider-Man series getting ready for the movie that's going to be coming out soon.
Can't wait.
You do have, in other comics, you see similar villains and superheroes that are created the same way. And this kind of, let's mix together the best traits from all these things and create the super human or super villain or to go up against our
Spider-Man's
superhero.
in the same universe as the mutants.
Yes.
Yes, he is. Mm-hmm. Yeah. That's why Mr. Sinister was. He was trying to, he was recruited by Apocalypse and then to make.
No, there was a whole thing. I don't think it wasn't Civil War. It was, or maybe a spinoff of that. I can't remember. I didn't read all the comics. They were doing mutants versus everyone else, essentially.
Avengers versus
X-Men? It wasn't X-Men. X-Men were a big part of it, but it was across all Marvel Universe. And so Spider-Man was technically a mutant. And so he was on that side, but he had his relationship with the Avengers. And it was a really interesting concept. And I was like, oh, how are they identifying mutants? And they were like, we identified the X gene. And that's not a thing. You get mutations on tons of different genes. And everything can be technically a mutation. I've got hazel eyes. That could be a mutation if you compare them to brown eyes or other eyes. So it's a little iffy. Stan Lee did an interview where he was asked, how did you come up with these concepts? And he came up with some of them. But sometimes writers would be like, hey, Stan, I think this would be really cool if this character, the way he got his powers, let's just say he was bitten by a radioactive spider. Yeah. And that sounds sciencey. Let's do it. Yeah.
Right.
Well, I mean, I was like the scorpion. Like I said, it's a natural predator of spiders. So we went a villain. Let's go with that. Rhino, it was like, oh, they got their tough skin like armor, which really it's just their skins is really thick in its composition. But it was like, oh, we can make a fusion chimera with a person and they can inhabit and this structural thing. But it was like the science, where it was at and what was known. There was a lot of handwaving between that and the actual factual. So.
But there are like hidden little sciencey nuggets within science fiction. One of the coolest things that I've learned doing all these deep dives is electromagnetic pulses of the heart. We figured out, we didn't figure out, but we had to eventually. But it first appeared in science fiction as an idea of restarting hearts with electromagnetic pulses.
Right. Yeah.
Do you think that's because someone tried it and then they were like, hey, word is that if you use electricity, it'll help you get in? Like,
no idea. Like,
which came first?
In Frankenstein.
Exactly.
It's like, then Frankenstein, that was all based on they were trying to revive bodies. So that was episode 1 of Rabbit Hole of Research. You can go back and listen to it, y'all.
Wait, I don't
We
remember
did a
this
deep
episode.
dive,
Was I
yeah.
in this one?
You
No,
weren't
you
on
were
that.
not.
Oh, I don't know.
That
I
was
don't.
before you.
Episode
recall
one's
this
the
one,
only
then.
episode without Nick. So,
You probably didn't even listen to episode
I know,
one.
he-
I
Did
did
you?
too. Joe forced me.
but
Okay.
yeah, we talked. It was a really interesting time where they were doing a lot of experimentation to revive bodies, and that understanding of electricity was there. And a lot of people think that Mary Shelley she was friends with all these because it was all this kind of elite kind of thing to do. You would have a dead corpse in your
It's
parlor,
a theater.
and then you would try
Yeah.
to revive them with electric shots, and they would move. Laws were created, like what happens because a lot of times the bodies they had were criminals that had been hung, and they said, well, so they made laws like, well, if the body gets revived, then they would get hung again. Like we would, it was like, yeah,
Would that stink?
it was
Oh my
That is
God.
hilarious. I love it.
Yeah.
The thing to do for the elite.
That's right, yeah.
Let's have one of these. Let's
Not
have
too
a
far
trip
off
together.
from how we are
Yeah.
now.
Come on over. You're going to watch us revive this corpse. And so, yeah, there was like these kind of scientists, and even it was like different people who they stopped doing that. And they were like, this is kind of dumb, but hey, this electricity thing is really cool. Let me experiment with
what
that.
are
Yeah,
some
right.
other things we could do?
But yeah, Mary Shelley would go to these events, and a lot of people think that that's where the inspiration for Frankenstein came from. So,
That would make sense. Yeah.
yeah. Hmm. Yeah. But yeah.
I
it's kind
still
of
can't quite get over how a monkey bit your face.
I like to just stick my face near
Oh,
monkeys.
what are you doing?
I was trying to
that's
get the rage
happened. I
virus.
was using a real example.
Oh my gosh.
Yeah.
I was trying to find the rage virus. What can I say?
Oh no, the rage virus.
What's-
Monkeys are just full of rage regardless. Terrible pets.
Yeah. So
What other villains you got for us, Joe?
got the lizard. We just watched the 1981 Spider-Man animated cartoon about the lizard, and
And he
Peter.
let out the other reptiles from the zoo.
He built a weather machine, the lizard, and he was going to turn New York into a swamp, and
I
he released
don't
all the reptiles.
Recall this one.
But it got resolved pretty quickly.
Yeah.
He was my favorite supervillain. I would love everyone to be lizard.
I know.
I stand with you.
So is that where
was his
the
thing.
plot for the Amazing Spider-Man came from then?
Maybe
Because didn't he try to go up in the air to make everyone lizards?
make everyone. Well,
Yeah.
then I think he's done a few things. Yeah, I think that was his
Devious-
idea.
That devious.
But he was one of Spider-Man's earliest reoccurring villains. Was the lizard? He was a surgeon, lost his arm in combat. He was distraught. He wanted to grow limbs, and so they picked reptiles, which actually reptiles don't really regenerate their limbs. And so
They didn't regrow it, but it
that
looks
was-
very different.
that's why it was one of the science things where they thought, because they will regenerate their tails, but they don't regenerate limbs.
Don't
per
some
se,
of them?
so. Salamanders do.
Oh,
so
that's what I'm
kind of
thinking
right.
of.
Yeah, lizards and salamanders they look similar, but they are.
And their
different.
limbs can completely regenerate,
That's right,
and it
yep,
looks exactly the same.
So it should have really been.
It should have been
salamander.
salamander. That's right. Which I
Yeah.
think there is a villain called the salamander.
Feel like there is. They might
Yeah.
be like a D-list villain somewhere named the Salamander.
Yeah, I think that was in X-Files.
Oh, was it?
Oh,
Oh.
you are correct. That was like early on in the
Yeah.
X-Files.
My husband has maybe watched X-Files all the way through twice at this point.
Oh my god.
Only twice?
And now we're gonna maybe
He's
get.
preparing for a third.
We might
know.
get a new X
The
Files.
new Ryan
right?
Coogler's got the new
Yup.
X-Files coming out.
I'm excited about that.
Yeah. The other thing with
Forget-
the... Oh, go ahead, Aaron.
Sorry, go ahead. You were explaining something and we were all interrupting.
No,
and then
no,
I
you're
had to
fine.
video
No, this
stem
is-
cells.
This is what
So,
happens.
you
No,
go
I was going to say the lizard. The other thing is that he can control the other lizards.
Right.
Like
he
that
was
was the thing.
sending
Yeah,
out
he
signals.
was sitting in, and then
right?
Spider-Man did something with the power plant
Or-
to disrupt
or
his
the radio,
electromagnetic frequency or something,
yeah.
and he was like, "Oh, I can't control it."
Is it my?
I think what would make more sense if it was like a pheromone thing.
Yes, that would. Yes, but no, that's
Was he tonguing the air the entire time?
no, he wasn't. What?
No.
Like Hannibal
I don't-
Lecter over
I don't
there?
think
Some
so.
fava
Lizards talk with
beans?
their tongue?
I don't think he did that one.
No,
Yeah.
I don't believe it
No.
as a lizard then.
No, but that would be a snake, a reptile. But
Aren't snakes reptiles?
they are, yeah,
All right,
but not a lizard.
Reptiles.
Okay, well,
What is a lizard other than a snake without arms?
That's true. Or no, other way around. But
Yeah.
Flip
they're
it,
both
reverse it.
squamates. Yeah, they're both squamates. They're very closely related, and snakes do have vestigial hip bones.
Boom.
Yeah.
Thank you.
That's exactly what you
Yeah,
were
that's what you said.
I
Yeah.
said that verbatim.
We're going to give you.
But going back to the lizard
Yeah, go
as
for
a
it.
villain, if he wanted to regrow his limbs, he should have been looking at stem cells.
Mhm.
And I think he
Yeah.
was just looking at genetics and not the
Yeah.
very base
Yeah.
cells. And salamanders can regenerate limbs because their
Mhm.
stem cells are still active. Humans, our stem cells deactivate. They're still there, but they're
Yeah.
not turned on. So current research to try and regrow limbs is like, how do we turn on these stem cells? How do salamanders keep theirs turned on? There are instances of very young children, like a couple days
Yeah,
old,
fingertips.
regrowing
Yeah.
fingertips.
Yeah.
Oh.
Mhm.
Their
Really?
stem cells are still on.
Yeah.
How long does it take to regrow that? Do they like?
I mean, the fingertips were small.
Yeah, it's not. I mean, especially in a little kid. That
We're
little
talking like an
baby.
infant.
Yeah.
I mean,
Yeah.
still
Yeah.
like a fingertip's a fingertip. It's
Yeah, a lot of and I was going to say a lot of that research is in the axolotls. There it is. Thank you.
Come on, Joe.
That's where. Hey, you
I was
know
just
what?
gonna let you flounder with that one. I know how to say
I'm just
that.
going to go.
word.
Well, say it then.
That's
Jump
because
in
you have
there.
a young child,
I
and
know.
all children.
But that's one of the model organisms for limb regeneration, and that this idea that Erin was just saying about that turned on. So they do have pathways. There's different genes they know that come
They're
on.
cool. Aren't they
And-
cool?
Yeah.
And to that point, humans have all those genes and responses, but they just don't. They ignore that. So if you lose a limb, it's like, I'm just going to make scar tissue instead of regenerate the limb. So there's pathways there, and it just says your body goes, nah, you know what? You had a hand, you got it chopped off, so live with the consequences.
That's really weird that we have the ability to, but our body just stops.
Yeah,
Right.
and it could be some evolutionary bits in that, right?
It would make
so,
sense to keep it going then.
well,
Like,
if you had it, if
If
you
it was
are,
happening a lot, I guess.
if you lose a limb and you have to keep going, how long can you stop and actually heal? So is it better just to form a scar, heal up, patch up, and then move on? Or are you going to really attempt to try to reform a digit or a hand or a limb that won't work properly or
And
won't?
how many Big
how long
Macs?
will it take so how long will it take
And
this?
it takes a lot
you
of
know,
energy
right,
too.
right,
and
right.
we already divert a ton of energy into keeping us warm and to our brains.
So that could be part of it. That it's more like the healing process. Let's just go ahead, cells, get this over with as quickly as possible, so that this person can keep moving, can procreate, and keep their genetic lineage strong and moving forward.
Wolverine could do it.
If Wolverine could. Yes, Wolverine is our.
Which we've already done the episode with him.
We
And
did do, and Deadpool.
Yep.
Yep.
So I feel like that's something that would be cool to see come back. Like if science was looking more into the stem cells to be like, oh yeah,
They are. No, people are studying that. There's a lot of folks
I would be
like
willing
so,
to cut off a hand to try this.
like
No.
in like lamprey. So lamprey is this little eel, and so I know people that work on that, but it's an invertebrate. It has a notochord that is like a primitive spinal cord, and it could be severed, and then it will fall to the bottom of the ocean and get covered with sand, and then sometime later it will regrow. It would make another functional spinal cord, or notochord is the term for it, and then it will live on, but it needs time to just stay inactive. All it's doing is just laying there and then healing itself and regenerating. So
What's
it's
it doing for the energy though?
it's just
It's
Like,
just
would it have
staying
to have
still.
to go ahead and eat
Right.
as much as it can before dropping down?
well, it can. You
It just-
can't predict when you.
It's a different
Yeah.
organism.
Yeah.
They don't have endothermy. So we are endothermic. We create our heat from the inside. It's inside therm heat. And they are not. They're ectotherms. They get their heat from the outside,
Right.
ecto outside. So they're not eating
Yeah.
as much as we are because they don't need to generate all this heat. You eat 3 meals a day, partly because you are an endothermic animal.
You really are.
Thanks. Is this an insult, Georgia?
I don't
You know,
It
just a
just
human in you.
sounded good.
But yeah, so that's there. Like I said, people are working on it, and I think it's since we know how it works in here, can we figure out? And we have all the bits. Can we just figure out how to turn it on? How can we switch all the levers on to get this to work? And
Can
to
CRISPR help?
do? This isn't, I mean,
hypothetically going into the embryo and then being like, all right, just keep this one turned on.
I think it's right. So I think with CRISPR,
Or would that,
CRISPR's
like,
job is to
grow
add
another
I
limb?
think it's to add in functionality. And what I'm saying is that you have the functionality, but it's not turned on. So
yeah, but if
then
you
the
have to
question
add
is,
something in to keep that functionality on.
but it might not be at the
on code. So
yeah, it might not be
theoretically,
at
I don't know
the
enough about it, but you could possibly splice in an on code.
you could, but it might not be at the genetic level. So it could be
I
at this
think-
epigenetic level where now you're having something external click off, which CRISPR in. So then could you modify that, but not screw up all the other developmental stuff, right? Because now we're talking about developmental pathways. So that because at some point in time, early on, all this stuff is on because you need to develop. And then it all turns off when you're done. Your body plan is formed, and now you're just filling out. Let's say filling out. And then something happens, an accident, and then now you got to regenerate. Now, all those signals, now you're telling it. So it's a very particular case that you need these signals to come on to now reform a digit, to recreate the body plan. So go, okay, this is what the body plans. Let's get these certain particular instructions out and build it. So that's kind of where you're at. So could you have CRISPR come in, get signals to say you've lost a digit and now turn on all this pathway? Maybe. Or is there another easier way? Just go, what is that on off? And can you control it via drugs or chemically? Can you just dip your hand into some solution and that will activate the cells and tell them, hey, you should be in this state, not this state? I don't know. And right, it's not my area of research, but yeah. So I'm not going to, CRISPR, it could be a way, but then what's the mechanism of on off? And that, I don't know that answer. So.
Willpower.
Yes.
Will.
Yeah, I use my lizard telepathy to turn my
Just will it.
That doesn't work. People always go to reptiles, like anything. Jurassic Park, reptiles. Spider-Man, reptiles. I think they're just cool. Reptiles
They are just
are neat.
cool.
Yeah, so
Yeah, they're
you
crazy
go
looking.
into
Look
it
at them.
and
With their little tongues going.
we need some Chianti and fava beans already. What's happening?
I
Yeah.
just like seeing snakes' tongues go.
But yeah, a lot of these organisms too are really cool. Like you eat like scorpion venom. It's like compounds. They have properties maybe anti-cancer, antibacterial, antifungal, antiviral. When I was diving in, it was like, oh, you read about the scorpion and
And
they
also,
can do.
it can also kill you, right?
There are. I think it's very few. I think it's 100th of the species. It's not that many. So there's thousands of species, and I think it's just in the 20 or 30.
It's kind
of
of
them
like
that can actually kill
spiders.
you, right?
Most spiders.
Most right. There's so many that they're not all venomous to you. So
Oh, I definitely didn't know that not all scorpions were venomous.
no, they have venom, but are they dangerous? Will they kill you, or are they lethal? I think that's the question, right?
Right.
So,
and it could be lethal to if a dog gets bit.
stung.
Versus,
Yeah,
Or stung
yeah.
versus a human.
that's right.
You know
Yes,
what I
yeah.
mean?
Metabolism's really different and things like that. So
I was not aware of how widespread scorpions were in the United States.
Yeah.
I look for reptiles and amphibians wherever I go, and I was outside of Portland years ago, and I flipped a rock, and under there was a scorpion, and I was like, hold on, it's in Portland. And I was talking to people, and we had no idea we had scorpions.
Wow.
And I looked on iNaturalist, and it was the second most northerly record of the western forest scorpion. And apparently that's when they're out anyway. So I talked to an entomologist, and they're like, one, it's an arachnid. No, you don't talk to me. And I was like, I'm so sorry. I am a fool. And then talk to somebody else. And they're like, yeah, we don't care. That's when they're coming out to roam around anyway.
I was not aware that they were not just in the southern hemisphere.
I think they're like
Well,
in
like-
every.
Or
I
They were
want to say
real-
they're like on every continent
They were
except
very
Antarctica.
they were very prevalent in Arizona.
makes sense.
Yeah.
Yes, that's what
Arizona, New
Like
Mexico,
you had
the
to check
desert
your
areas.
you have to check your shoes.
Don't you use black light to do that?
You can. So that's a fun thing that scorpions will fluoresce. Their exoskeleton will fluoresce under UV, under moonlight. It's really interesting that they have this fluorescent compound.
Now, I don't think Spider-Man has tried this.
Yes, that would be the way to look for scorpions.
It's
All right, let's
very
turn on
easy.
the black light.
And he can't commit any crime. It's like, who's here?
Go to the nightclub and
You just see him glowing. I mean,
I just think a man-sized creature with a long, oversized, misproportion. I think that's the other thing. The scorpion in the comics, his tail is much larger than per body area to a scorpion. I think it's a
you
little.
think it'd be hard to hide?
I think it's really hard to hide. Yes.
I don't know.
Doesn't he wear a trench coat and is kind of be walking around?
Sometimes, yeah.
He's
See, and that's when you need it.
Sorry.
That's when you need
Don't
to
trust.
know.
Don't trust those.
It's not like they live. He got glasses. Put on these glasses, you'll see the scorpion. No. Yeah, cool. That's cameras. We
Like
can
how
do it.
it was incredible how Doc Ock also wears a trench coat and his backlight's flat.
Oh, right. Yeah.
A lot of people like trench coats.
Yeah.
They're
Trench coat.
convenient.
I don't trust
Yeah.
anyone in a trench coat.
Trench coat.
Benny Hill.
I don't trust them.
You don't trust Columbo anyway.
Yeah, Columbo did, but I trust Columbo.
Columbo, yeah.
It's either a supervillain or a flasher, and I don't want to see either of them.
Or Columbo.
Or Columbo.
Could
Again,
be both.
still
Supervillains
stands.
Could be. Flashers are not considered supervillains.
I wouldn't consider supervillains.
That might be just a side gig.
Yeah. I don't know if they're be villains.
Perverts, not
They're not even
villains.
perverts. You could just have a trench coat on with nothing on underneath.
But if you're opening
Are
it
you, are
up,
you, hold up. Please hold.
is
What's
this
this
something
gonna do,
Joe,
guys?
is this something you've tried? Like, you,
No, I didn't say, once again, I didn't say I tried it. I'm just saying that. Why do you, all right.
Why are you?
Okay.
Because
You can wear it without opening it. Of
Right,
course.
exactly,
No
yes.
one's judging you, Joe.
You
But
can
then
go out.
you're not a flasher unless you flash, right? If you keep the trench closed and nobody knows you're
I guess.
naked
I
under
was
it,
just saying
you're
that
not-
you're not necessarily a villain.
does Joe? Do you have your own special trench coat?
If you're a villain. I don't own a trench coat. I'll tell you.
Georgia can confirm or deny
As
this.
far as I know,
I don't know. I know. No. I always think of Invasion of Body Snatchers. I wonder if they
Donald
were. I wonder if they
Sutherland,
were chimeras because they became the alien, like the thing. I think are they chimeras? They take over. They take over every cell, so they no longer are chimeras.
So
I think
you're now both.
But
yeah.
with the Invasion of Body Snatchers, you get infected.
But
by
I
the-
think
the
they're
space
similar
pod.
to the Thing where they take over.
But,
that.
I mean, why?
But that, no, you're
Yeah.
right because they keep.
Yeah.
They stay
Yeah.
a human.
You stay human, they just infect you. So are you now a pod person? But that's because they've infected, and that's one
and
of the
is
things
there
too.
any
They
way
don't.
back? Is there any way
They
back?
a lot of research. When I was looking this up, there's a lot of rules and some of that they won't like neuro chimeras. So with the pig hearts or pancreas or other organs, they're fine. But when you start saying, oh, we're going to make chimeric, that sounds like a spice or something.
I was thinking limerick when you said chimera.
with neuronal tissue, brains, then that's kind of where the big line is at. Don't cross this line. And a lot of that's because of consciousness and things like that.
Oh,
So if
really?
you make a pig human brain, and then is it human? Does it have human thoughts or emotions?
That's interesting.
And then,
yeah.
And then you put it in a human body. Will it start
Oh my
talking?
gosh. Or will the pig just
Oh, I love this idea.
That gets into interesting where your memory's stored, and if you take some of that, but then you replace it, will you remember being a pig?
right?
Or will you just start reverting back to your pig-like self?
Well, and like I said earlier, that the pig to human ratio in these chimeras is small, so you don't have a lot of human cells there. So what cells are important? And I think there was an episode I saw, Flowers for Rhino. It was the Flowers for Alderaan, the Daniel Keyes story about the mouse who was given super intelligence, and then they gave it to a person who had learning disabilities, and then he became super smart, and then they realized that I don't want to spoil it. Maybe this is a pretty old story.
It's
But-
a very old
yeah,
book,
yeah,
but it's
yeah.
so good.
I don't think
It
I'm
is-
aware of this.
it
You
is
not read
really
Flowers
good.
for Alderaan?
I don't
Yeah.
think so.
yeah. You
It's,
should-
and
If
there's
you
a
want a
All
good
right.
cry,
Yeah.
and there's
Yeah.
a
Really
good
good
and
cry.
the movie is
And the movie,
good,
like
A
too.
the-
commercial can get me to cry.
Yeah.
Hold up.
Yeah.
It's
Yeah. So,
I
so-
real
was sobbing
easy.
reading the short
Yeah.
story version.
Yeah,
That was a
it's
text
really
book.
good.
did you read the novel? They lengthened it and they
I
made
haven't
the
read the
movie.
novel.
Yeah, it's
I
pretty
read
good. Yeah.
Like a 40-page short.
Yeah, that's right.
short
Yeah, it's like a
story.
novella. Yeah. Yeah.
Yeah,
And then
the-
the movie was
The
good, which
movie
is
I thought was good.
based on a novel. So they extended it more about the relationship. So they just stretched it out a little bit. So you really felt bad at the end. But yes, it was really good. But there was a comic run, Flowers for Rhino. And that was his whole thing. 'Cause I think part of his thing was to get super smart. That was that was the goal. Like, it's always like some goal. Like, just like, you want to do this thing. Is it revenge or some Faustian bargain? You're going to get super smart or live forever.
It's always a bargain, unless you're a super villain or a super scientist, and you're like, I need to control the world.
Oh, okay.
Is that you, Joe?
Yeah, that's.
I think I did a spot on Joe in
And your
impersonation.
trench coat? I think you got a trench coat.
I don't.
A science coat's a little bit like a trench
A
coat.
lab coat. Yeah.
It's true.
Yeah, they are indeed. It's the scientist
And they
trench
probably
coat.
frown upon flashing.
Are you allowed to go without
There
clothes
are
underneath
there's
those
the
coats?
safety. There's like the safety videos. You're not supposed to. Yeah, you got to wear pants and socks and closed-toed shoes. It's a whole thing.
That makes sense.
We'll cover that in lab safety.
Well, that's right. We had that
I
episode.
worked at a lab in New Orleans, and it was so hot outside. But then when we got inside, it was so
Right.
cold, and we
Yes,
only
yes.
wore the lab coats if there are people who don't want to give us money. Were around, so we had a separate drawer for every person, and it was just full of cardigans and sweaters and closed-toed shoes.
The same thing and I went to grad school in Arizona State University, so it was pretty warm. So we wore shorts and t-shirts. Yeah.
So,
But yeah, I
question
think
for
that's-
everyone. If you can splice your DNA with one animal, what would it be?
Ooh.
Joe, you want to start us off?
No, I don't want to start off. You've been thinking about this for a while. No.
What? No.
You and your trench coat.
I think I'd go axolotl.
You go.
Yeah. I want the
And
axolotl.
Why?
did you think of that before or just since this conversation?
Oh, it's been a long running.
Oh, so you've been thinking about
Yeah.
that for a long
What,
time.
what are you going to do as an axolotl? I mean,
Going to become a superhero and possibly super
doing
villain.
what?
I can lose limbs, Joe, and regenerate
Yeah,
them.
but that's just a part of it, right? I don't. What else?
I'll deal with everything else
Nothing.
later.
I don't know.
That's the only part I want of it.
Okay,
I can regeneration. Come on.
Erin, what do you got? Anything?
I don't know. I was gonna go salamander, and then I was like, oh, hold on a second. Let's think a little harder. I don't know. If I was like a full other animal, I would choose bald eagle because then anyone who shot me would be persecuted. And I would go be avenged.
Are you
I'd be
under?
a very safe
Yeah, right.
bird.
Are you in the threat
I
of being
know. Yeah.
shot?
What are you doing? Like your secret agent in your trench coat? That's
But I don't know if I
another
would,
person
like,
I
be
don't trust in a
spliced.
trench
Are
coat.
you also in? Do you lose a lot of
I
limbs?
will.
I know. Yeah. Well, Reese from Terminator. Oh, Mantis.
Mantis
Oh,
shrimp. Mantis shrimp.
Mantis
so you
shrimp.
can see
Oh,
all
yeah.
theirs.
Oh wait, is that not the one that does the snapping? That like
Yeah. That could be my power. I wanted it for the vision. I wanted to see all these different colors.
Yeah. Yeah.
I thought you were like, I want to snap
Yeah.
at people and make them hurt.
Interesting.
I think
I want
maybe-
to be like really psychedelic life.
Yeah.
Let's see what's going on.
I think maybe a cat.
Cat.
So you could just lay in the sun all day. You can do that now.
I know. Yeah. I don't.
Just go
what's-
outside,
what are we getting
Georgia.
from the
Because
cat?
cats are cool.
Cats are cool. Not a dog. Cat.
I was actually thinking of pig because I really like
yeah.
pigs,
Yeah.
and they
Yeah. A lot of
And we talked a
biomedical
lot
research
about
you can go.
pigs,
Yeah, we did
so-
talk a lot about pigs. Yeah.
Jotham.
Got it there.
You can-
Okay. I don't know. That's a weird question. I'm
All
thinking-
my questions are
I'm
weird.
still thinking about the trench
well,
coat.
if you needed to fight Spider-Man, what?
If I had to fight
Why are
Spider-Man.
you fighting
I
Spider?
know, why am I now a
Lady
villain?
Pig?
Because it's like
Why
most
do I
of
gotta
these
be the villain?
villains
Yeah,
are the villains of Spider-Man, so maybe you could think of it that
that's
way.
true. Yeah, but like we talked about, a lot of these mutations don't work the way we want them to work. So I don't know. What am I getting from all this? Maybe I'll be an octopus. I'll be like the real Doc Ock.
I could see that.
I can have more arms, maybe.
Yeah.
There you go, and you're like doing stuff, and I can squish myself into little spaces.
You can
could probably-
camouflage.
Camouflage, that's right. Got the skin. I
You
can
probably
do all
have
that.
to get
yeah.
a custom-made
Yeah.
trench coat.
Yeah. But you know what? I'm gonna go. Okay, I'm gonna go with a plant. I would like to do photosynthesis. I think that'd be cool.
That's a
Interesting.
good
Yeah,
adaptation there.
Just go. Yeah.
So then
maybe I'll
eating
go
you
there.
would be vegetarian.
No, you can't eat me. I'm part human still.
Well,
I'm
yeah,
just,
if you're
right.
a plant and you eat, aren't you cannibal?
Am I a plant? Yeah, right. Maybe I can't eat plants. Maybe I can eat you then.
But also, you
Okay.
have photosynthesis. You can make your own food.
That's right. Yeah. You know what? I'm set. I don't have to eat anything.
No Big Mac.
I can just go. I can have a Big Mac if I want a Big Mac with my lettuce, eating my people.
Eating my
Hold the lettuce.
Yeah. Just mayonnaise. Give me that.
All right.
Well-
Well,
Thank you so much for being here with us. Do you have anything to plug?
Or add to the conversation if you've some research bit that we didn't get to. You want to just mention it right now really fast. But yeah.
I think we got to everything except for xenografts. That was
Xenografts.
interesting
All
to
right.
go. It's grafting animal parts from one species into another.
Okay. Yeah. Yeah.
But
Yeah.
I don't have any
Yeah.
plugs for any future events. I'm doing some maybe some trivia with the Science Association,
Cool.
but I haven't fully gotten that sorted out. I'm going to be at MarsCon next year.
Nice. Yeah. I'm hopefully be there.
cool.
And I was going to say with that is not, if you put pig heart into humans, wouldn't that be grafting an animal organ into
Oh, you're
another
right.
one?
You're
Yeah,
right. We did.
so,
I just
yeah.
didn't get to say
Yeah.
it.
You didn't get to say the
I learned
fancy
that
word. Yeah.
today.
It's such a cool word.
You got to say the fancy word. That could become the word of the season, like
I don't know.
extremophiles.
It's pretty cool. Yeah.
Georgia didn't actually say it this
Well,
this episode.
Well,
yeah.
Joe did.
Just because he
There you
was
go.
bringing it
Yeah,
up.
right. Well, thanks again for joining us. It was a lot of
Oh, thank
fun
you,
and
Frank.
talking about chimeras and Spider-Man and then all that. But you got me, Joe.
You got Nick.
You got Nick.
Georgia.
You got Georgia.
And we went down some holes.
Some different mixed up holes. Half holes. Chimera holes.
Bye-bye.
Stay curious. We love y'all.